morning, everybody. I wanted to first thank ARM for the kind invitation to have us here. We're one of the new kids on the block, and we're very proud to be a part of the family, as it were. So Homology was founded about a year and a half ago, so we're pretty much new kids on the block, but with a very exciting platform that encompasses or bridges gene editing capability and gene therapy capability. And the way we look at this field right now, we're at a critical point in time where gene correction can offer essentially a cure for patients with rare genetic disorders who essentially have a death sentence hanging over their head. And this technology is really based on a novel family of AAVs that were found isolated from human CD34 cells, so naturally occurring AAVs. That have the ability to mediate either gene transfer or standard gene therapy or AAV-mediated gene editing or gene correction. And we can conceive of doing this as an in vivo, highly specific, highly efficient process that is without the need for exogenous nucleases. The platform can be used to address a broad range of rare genetic disorders, either through gene therapy, where we think that's going to represent a cure, or through gene editing or gene correction. There we go. So this slide really, I would say, puts out the underpinning tenets for the company. It's a single AAV nuclease-free platform that can mediate in vivo gene editing. And as you know, most gene editing approaches now are nuclease-based, starting with ex vivo and then moving towards in vivo because delivery represents a real challenge. The idea for the company from a corporate strategy point of view is speed to the clinic. Initially, we'll do this with a gene transfer program, which I'll get into in a few moments. Again, the way that this works is not something new. It's been known for over 20 years that AAV can mediate HR-mediated gene correction. This family of vectors seems to do this at high enough efficiency that allows us to conceive of an in vivo approach versus an ex vivo approach. We have core intellectual property license from the City of Hope, from the lab of Dr. Saswati Chatterjee, and we have a single IP estate, so we're in good shape on that front. And as Jeff just mentioned, I think anybody from Shire who's not working for Avro is now working for Homology. So it's a team with a lot of experience in developing from the bench all the way to commercialization rare genetic diseases. Many of them were ERTs, some of them not. But with Albert Seymour as our CSO, who's in the audience, who was just on a panel earlier today, Sam Rosti as our COO, and we recently had Brad Smith join us, who we call the adult of the group, who has a great deal of expertise with small emerging growth companies. So it's a real strong team with a lot of experience that combines gene therapy expertise and rare disease drug development expertise. So how do we think about this platform? It's a platform that has two arms to it, and we really let the disease biology dictate which approach we will take. So on the gene editing side, on the left-hand side of the screen, we really feel we have an advantage at being able to deliver wild-type normal genes in exchange for mutated genes, and that's really the basis of how this works in terms of homologous recombination. We have the ability to correct these mutations in this way, add entire genes for diseases where there's hundreds of mutations literally that cause disease. We also have the ability to knock out disease genes, but that's something that right now we feel is not high up on our priority list because we feel we have an advantage in gene delivery. Now on the right-hand side, these vectors that were isolated from CD34 cells have really good biodistribution, and we've shown this in non-human primates now to multiple tissues, including the ability to cross the blood-brain barrier. So we feel we can go after CNS diseases where there's a huge unmet need. So the way we're thinking about developing the company, creating a lot of value, but also, more importantly, going after the key mission for the company, which is to actually cure genetic diseases, whereas before we were used to drugs that could merely treat symptoms of genetic diseases, is to really move forward with a gene therapy approach first, and de-risk the use of these novel vectors in humans. And we are moving forward in development stage with the inborn error of metabolism disease as our lead program, and then use the learnings from that to move forward the gene editing side of the platform. So disease areas we're considering are obviously CD34 diseases where you're going to need editing because these are a cell compartment where you have great cell turnover. So the hemoglobinopathies are a good example. Lysosomal storage disorders, particularly in the CNS because of the ability of these vectors across the blood-brain barrier. And we have very strong preliminary data in cystic fibrosis where an editing approach is probably the best approach there, as well as DMD. So from a very high level pipeline-wise, we already have a candidate, development candidate for this inborn error of metabolism disease that we're moving forward doing non-clinical work, pre-IND work on. And then we're kind of letting the science dictate which of these programs will emerge as our lead editing program. Just want to spend some time on this slide. It's kind of a data-heavy slide, but we have spent a lot of time in making sure that the way we are doing gene editing or gene correction and how we're editing genes is indeed highly specific with very little to no off-target. So on the left-hand side, you see the way that the DNA constructs of these editing vectors are set up with homology arms that flank the correct gene sequence, and we can use a fluorescent probe here to assess the phenotypic percent correction or addition of a gene to the genome. And you can see we can do that by flow. But very importantly, on the bottom right-hand side of the slide, you see that we have evolved a number of assays to determine how precise is the addition of this gene to the genome. And I think that's one of the key attributes of this technology, that HR is a highly precise DNA repair mechanism versus NHEJ, or non-homologous end joining. And we've evolved a ddPCR, or digital droplet PCR assay in the lower right-hand corner to really be able to assess on a per-cell basis what percentage of alleles we've edited. And in further work that we presented at last year's ASGCT, we have shown that the off-target rate of this process is as close to zero as you can get. It's within the error of the sequencing assay. And this is one of the hallmarks of HR, but it's something that we wanted to firmly establish, and I think it's something that the editing companies need to really focus on and be very, very, I would say, rigorous around from a regulatory point of view, is how to measure off-target. So this is not predictive algorithms that we're using. This is actual measurement of where this insert goes, and we know precisely to essentially the nucleotide where it is in the genome. And so we feel that's a very important hallmark of this technology. So from a point of view of this AAV-mediated gene editing, we feel like it's very, what we would call developable. We can be moved to the clinic relatively rapidly. We feel delivery is a key differentiating point for us because we're using a single AAV with excellent tropism to tissue. So this can be used for gene transfer or gene editing. The on-off target we just discussed, where we feel like we have an advantage here. The HR in vivo efficiencies are high enough for us where we can contemplate going after diseases where you can actually in vivo correct. And we've been, I think, very careful about which diseases and letting human genetics tell us where we need perhaps 10% expression of the normal gene in order to get that correction. And that's been known in the genetic rare disease field for quite a while. You just need to get to a certain percentage with key diseases, particularly liver diseases, and that will result in a normal phenotype. So because it's a single viral vector manufacturing, which we're highly focused on as well, as Jeff mentioned, it is a key thing that we've lived with in our years at Shire on the protein side. It's a major issue and very key attribute that we have focused on right out of the gate for the gene therapy side. So because it's a single vector, we feel like there's a pathway already in place for us to follow to make sure that this is not a rate-limiting step for development of the platform. And then on the commercial side, having a single injection in vivo therapy really leverages the platform and we feel allows the transformation from basic research, basic science, non-clinical, clinical, all the way through commercialization. Just a little bit of data for you. These are two different animal models, two liver inborn errors of metabolism diseases. On the left side, what you have is a saline-treated animal model. So it's an animal model that's missing a particular enzyme that leads to the increase of a substrate, which is toxic. And you can see the untreated animals are sitting there at 2,000 micromolar. With a single injection of our development candidate here, you can bring this down all the way to normal levels within 1 week. In terms of durability of the response, here we're showing 3 weeks, but this data now is out to 3 months in this particular animal model. On the right-hand side is another inborn error of metabolism disease, again, where there's an enzyme deficient in this mouse model. Single injection of this development candidate brings those levels of the substrate down all the way to normal levels. So we feel like we have very highly developed, highly involved vectors that can be moved into non-clinical studies with these diseases. We've also, in addition to the City of Hope technology, which forms the core of the company, have in-licensed Ben Devvarman's technology from Caltech. And you can see this is from a paper published in January 2016, Nature Biotechnology. The ability to, and he used a screen for optimizing peptides or capsid peptides that enhance the ability of AAV9 to cross the blood-brain barrier. AAV9 crosses the blood-brain barrier pretty well on its own, but you can see that there's a 40-fold increase in the ability with these peptides as part of the capsid of AAV9 to improve the ability of these vectors to cross the blood-brain barrier for CNS diseases. And so we feel that grafting this technology on top of our own AAV technology will lead to the best-in-class vectors that can be given IV, but can get enough into the brain, enough of it can cross the blood-brain barrier where you can get the levels required to actually go after many rare genetic monogenic disorders in the central nervous system where there's a huge unmet need for patients. We have a strong IP position. We have composition of matter on 15 of these novel AAVs that were isolated from human CD34 cells. We also have 95% composition of matter around each of these, in addition on the right-hand side to the Caltech technology. So we feel that the marriage of these technologies is going to lead to a platform that we can use to develop numerous important medicines for patients. We've had a busy 2017 as we've kind of gotten our stride or hit our stride as a team. We're in IND-enabling studies for our lead IAM disease. Goal for this year is to have one of these gene editing vectors come to development candidate status. We've continued to expand IP and also build on the existing IP that we have, expand and progress our CNS programs. And we've made a huge amount of progress on the manufacturing front in terms of building out that capability of the company, which, as you all know, is extremely important. So I think we're set up very well in 2018. I just want to mention that we're very proud to be part of the new kind of wave of technologies and companies that have as its central core patients who are living with these diseases. We feel that we are really one of the companies, along with many others in the room and at this conference, that are really focused on the patient and having the patient be at the center of everything we do. So I wanted to thank you all very much for your attention and have a great conference. Any questions?